Hi everyone. So I decided to create a microbe that creates pathogenesis in the United States in that it promotes insulin resistance and type 2 diabetes. This is a topic I'm really interested in, so that's why I chose to make something that perpetuated this disease. So it is called Hepatobacter ketovibrio. Essentially, it is a gram-positive Vibrio bacterium that resides near the liver, and it does most of its damage there in that it halts ketogenesis. So it prevents ketones from being produced. And it does this so that the body stays in a fed state. They constantly have glucose in circulation and thus store more fat and do not use fat as a primary substrate. Additionally, it causes symptoms of laziness and drowsiness, lethargy, and it is harmful, clearly. It perpetuates insulin resistance and type 2 diabetes. Some of the structural features that you can see in my drawing right here are that it is a unicellular organism. The purple layer here is peptidoglycan cell wall with lipoteichoic acid and teichoic acid. It contains an S layer for attachment, fimbriae, so that it can more readily attach to liver cells. It also contains granules, which you can see are the blue little balls in the middle. They store enzymes which can help dissolve ketone bodies. It has a flagella, which allows it to propel itself in the human body. It also contains an outer membrane. A little bit about the genetic information is it has a chromosome with, so it is a chromosome with 1.6 million base pairs. It has plasmids, which those are those green circular objects in the center of the bacterium. It has an operon called the ketooperon. So something interesting about this is that genes are activated when someone is in a fasted state and their body tries to produce ketones. And this operon synthesizes and releases enzymes that halt or degrade ketone bodies. So it prevents the ketones from actually being formed. It has a sex pili, which allows for conjugation, so that kind of mode of reproduction, and it can also be involved in mutations. The primary energy substrate for this bacterium are lipids, because they want to consume all the lipids so that people aren't able to use the lipids for fuel. They also can reside in the human body, 37 degrees Celsius. They are facultative acidophiles because they can also reside in the stomach, but they also can go into the small intestine, which is a higher pH. Mesophile because, again, living in the human body. They also require micronutrients such as calcium and potassium. A little bit about the specializations of this bacterium are that they can form a biofilm on the mucosal layer of the GI tract. The bacteria additionally can aggregate on expired candy, which is kind of an interesting thing in that a human can ingest this bacterium, and that's kind of how it gets into the body and can do its virulence in that way. They primarily hide out in liver hepatocytes, and I don't know if we learned, but in physiology we know that macrophages like to hide out in Kupffer cells. And they can also reside in the stomach and small intestine, like I said before. The enzymes that work to dissolve the ketone bodies are called ketone dissolutionases. They work to dissolve the ketones. They also have a lethal and edema factor, which again contribute to the pathogenesis. Some of the components of the immune system are that they have PAMPs, so pathogen-associated molecular patterns. But they can do some avoiding of the immune system, and they do this by avoiding the phagosome before lysosome fuses, and they can hide out in those cells near the liver hepatocytes. They also have an M protein on the fimbriae, which prevents them from being engulfed by the macrophages via phagocytosis. And finally, they have a peptidoglycan cell wall, which prevents osmotic lysis. So again, allows them to remain in the body longer to create more pathogenesis and create an environment that is prone to insulin resistance, damaging those receptors due to increased glucose in the blood, and finally resulting in potentially type 2 diabetes. So that is the overview, summary, and a bit of the virulence mechanism pathology regarding this new bacteria.